So just to introduce myself, I'm Professor Peter Barlow. I'm Chair of Immunology and Infection at Edinburgh Napier University. And my research interests are really centered around understanding how the immune system interacts with pathogens like bacteria and viruses, and how we can use or exploit that knowledge to design new drugs that are based on components of the immune system that you could then potentially use to treat infections with bacteria and viruses. So for the past 10 years, I've been interested in respiratory viruses like influenza, rhinovirus, and what we've been doing is trying to understand how a family of peptides, which are very small molecules, which are a key part of the innate immune system, actually interact with viruses and what sort of antiviral activities they exhibit, and whether we could use the structure of those peptides to develop new drugs that would actually be delivered to patients and inhibit those viruses. So the PlusPEP project is an emerging future spinout of Edinburgh Napier University. Our aim is to become a well-being company that manufactures and sells products that enhance our natural immunity against rhinovirus. And rhinovirus is a primary cause of the common cold. So the challenge is that there's currently no cure for the common cold, and treatment is frequently with cold and flu remedies that treat the symptoms of the cold, but not the underlying cause. And the underlying cause is most frequently a rhinovirus infection. And it's a very prevalent infection. So in the UK, there's about 380 million rhinovirus infections annually, and that results in about 26 million sick days per year to the UK workforce. So on a global scale, the impact of that is around $40 billion in lost working days. So our approach is really to target the underlying cause of the common cold, which is most frequently a rhinovirus infection. So the PlusPEP formulation is essentially designed to enhance the production of a small antiviral peptide that's called LL37, and your immune cells essentially produce LL37 when you get infected with a virus. And we've shown that it's a key part of the immune response, and that the peptide has really potent antiviral activity against rhinovirus. So we aim to take this research into the marketplace by making PlusPEP the first non-prescribed over-the-counter medicine that will directly target rhinovirus through enhancing the functions of the immune system. So just to tell you a little bit about LL37, so LL37 is a member of a family of peptides called catalysidins, and most living organisms on Earth have some form of rudimentary peptide-based immune response. So we've been working as a group on LL37 for about 15 to 20 years. I first got interested in it during my first postdoc at the University of Edinburgh, where I worked with a chap called Professor Donald Davidson, and he introduced me to what LL37 did. And at that point, we knew that it was very antibacterial and that it had the capacity to change the way in which the immune system works. We secured some funding to look at the antiviral properties of LL37 towards influenza virus. I moved to the States. I worked for the Centers for Disease Control and Prevention, CDC, and continued working on LL37 and influenza. But when I came back to Edinburgh and started at Edinburgh Napier University and I established my own group, we started working on the ability of LL37 to exert antiviral effects towards rhinovirus, and that work was funded by Tenovus Scotland and the Chief Scientist Office. So what we've been trying to do over the past 10 to 15 years is understand how LL37 works, how it interacts with viruses, what sort of viruses are susceptible to its action, and whether or not we could actually use either derivatives of LL37 to treat viral infection as new drugs, or whether we can look to enhance the production of LL37, the natural production of LL37, as a possible antiviral strategy. So one example that I would give is that vitamin D is a very potent inducer of LL37. So that's why it's really important that people keep their vitamin D levels topped up. And essentially what's been shown in some infections, that LL37 plays a really critical role in the early stages of the infection. So anything that we can do to enhance production of LL37 will hopefully translate to a product like PlusPEP, which can be used to target viral infections. It is in really early stages at the moment. We haven't done any clinical trials. All our data is from laboratory-based models, but it's an exciting project, and we're hopeful that we can take it to the next level. So in terms of taking what I would call the peptide from the lab into the clinic, the process of essentially working with the peptide was very academic. The aims of doing the research were to advance knowledge about what the peptide did and publish that knowledge in peer-reviewed academic journals. Edinburgh Napier University is very keen to support emerging spinouts, and the university's had some success recently in some of its cyber-based spinouts. So after going to a workshop, which was run by the Royal Society, academics were encouraged to think about their research in different ways and whether their research could be used to generate intellectual property that might have commercial value or spin-out value. So I sat down with my collaborator, Dr. Craig Stevens, and we kind of thought about how we could do something completely different. So we'd been very focused on studying what LL37 did, how it interacted with viruses in the lab, how it interacted with different cells of the immune system. And we sat down and had a coffee and thought, well, is there something that we can do around promoting production of LL37, and could that translate to a new way of treating infectious diseases? So we did some experiments in the lab, some very rudimentary experiments, and we found that when we treated cells with what would eventually become the PlusPEP cocktail, that they were much better at killing virus. So then we spoke to our research office and said, well, you know, this is something that we could actually sell as a product. It would, you know, it would need to undergo clinical trials if we wanted to market it as a drug. But we feel that we're at the very early stages of something that could have commercial value. And they said, well, why don't you patent this idea? Why don't you patent the use of some of the ingredients of the PlusPEP cocktail for treating rhinovirus? So we applied for and were awarded a patent, and we've subsequently filed that patent in different territories around the world that might have commercial value at a later stage. We've entered several industry, or I guess commercialization related competitions, like the Converge Challenge, which is specifically designed to support early stage university spinouts. And we got down to the finals of the Converge Challenge. We unfortunately didn't succeed because we had some absolutely remarkable competition, but that was a brilliant learning experience for us. And I think all through the journey, we've been supported by a chap called Robert Goodfellow, who's one of the university's Royal Society of Edinburgh Entrepreneur in Residents. So he has been helping us through this process of commercializing this idea. He's our CEO designate. So I think for academics, the flipping the switch and thinking not just about publication of data and publication of research findings, but actually thinking about how those findings could be used in a commercial context. It's sometimes quite a challenging thing. I was absolutely terrible at it and still am, but I think with the support that you can receive from your university and people who have done this type of thing already, then there's so much scope and potential for researchers in Scotland to generate ideas that can actually change people's lives. And that's the most exciting part of the process. So it's a very nerve-wracking process, but it's a fantastic learning experience. I think collaboration is absolutely critical for many active researchers where they are trying to find a rapid solution to a very big problem. And, you know, for us, when we're trying to tackle respiratory viruses like rhinovirus and influenza that affect millions of people worldwide, that's very challenging to do by yourself, but a lot of value can be gained by collaborating with people that have expertise in areas that's complementary to your own. So I've worked for a long time with Craig Stevens, who is, I think he would describe himself as a gut scientist. So he worked on inflammatory bowel diseases, but he also worked on cell survival and cell death. And we kind of started working together because I was very interested in cell death from the point of view of viral infection, whereas Craig was interested in cell survival during inflammatory bowel disease. And our research interests meshed. And from that collaboration, we've been able to accelerate the work that we're doing much more rapidly because he has some extensive expertise in one area and I have experience in another. So for me, it's been a very good experience in terms of my own development, but also in terms of the work that we're pushing forward. And that's not just to mention, you know, I still collaborate with investigators from the Centres for Disease Control and Prevention, with people from the University of Edinburgh, Imperial College London. When we were doing this work, the University of Oxford very kindly helped us image the virus and find out what LL37 was doing to it. So all of these different things, we can push our research forward much more rapidly. And it's always really exciting. It's brilliant having a good collaborative network, people that support you, people that you can bounce ideas off of, someone who can help you out when you're not sure where to turn. So for me, the value of collaboration cannot be understated. And we've just finished a piece of work with some collaborators in Jakarta in Indonesia. And that was a remarkable experience for me because the investigators that I collaborated with in Jakarta were world-leading virologists, and just to see the level and the pace that they work at was absolutely remarkable. They were just so supportive of the collaboration and the work we were doing. And I'm hopeful that we can continue that collaboration, that we can continue to secure joint funding, which is why I think grants that offer the opportunity to collaborate with investigators at institutions around the world are so valuable. And I hope that type of funding continues and actually increases. I mean, we have a lot of next steps for PlusPEP. So in terms of the commercial opportunity that we have at the moment, there are no direct competitors for a product like PlusPEP that treats the underlying cause of the common cold. So our next steps in that respect are that we need to undertake product safety testing. We need to demonstrate that it has efficacy in vivo in patients, and that's absolutely critical to the success of PlusPEP. We've done a regulatory review, and for us, we know that PlusPEP fulfills the definition as a medicine in the UK, in Europe, and the United States. So we have to obtain MHRA approval, which is the Medicines and Healthcare Product Regulatory Agency. And we do that so that PlusPEP can make robust claims about the safety and the efficacy of the product. And then once we've done that, we would intend to launch PlusPEP through different, to different markets as a medicine. In terms of treating other diseases, we feel that the approach that we're taking, enhancement of antiviral molecules within the immune system, could have value for quite a wide range of viral infections. So it's our intention as another step forward to investigate that, whether there is value in either the same PlusPEP cocktail or a similar or more targeted cocktail that enhances different proteins of the innate immune system. And we could look to see whether that would be of value for treating an infection like COVID or influenza or another viral disease that we haven't looked at yet. The main thing for us is that we need to get more funds in. We need to secure more fundamental research grants. We need to secure investment that will allow us to do the clinical trials that we need to do to satisfy the MHRA approval process, and then get PlusPEP in people's hands as quickly as possible, so that they're able to benefit from the technology. It's going to be a long road, but we feel that we're making good progress. We've had fantastic guidance from Robert Goodfellow, and we just need to make it happen. So we'll be working as hard as we possibly can to get PlusPEP in people's hands as quickly as possible. So LL37 is a really exciting molecule because it has so many different functions. It can inhibit viruses within minutes. It can damage bacteria within minutes. It can change the way that your cells respond to an infection. It can alter inflammatory pathways. It's got lots of different functions within the immune system. And part of our research is focused on understanding how the structure of LL37 actually makes it behave in the way that it does. So what we've been doing is playing about with the structure of LL37. So through my collaboration with CDC, we've been looking to see whether we can make small modifications to the structure of the peptide, which would make it either better at killing viruses or more resistant to degradation if it was given as a medicine, because there's a lot of barriers to turning LL37 into a drug. It's not just a simple case of synthesizing it and giving it to people. Unfortunately, it's quite it can be quite fragile, and in some environments it doesn't work as effectively as we'd like it to. So what we're doing, as well as the PlusPEP project, is trying to understand how we can play with the structure of LL37 or modify the structure of LL37 and see whether we can make it a better version of itself. And we're doing that in a range of different viral infections. So we're currently still working on rhinovirus and influenza. We've just finished a project with collaborators in Jakarta on dengue virus. We're working with them on Zika virus as well. So there's lots of scope for the molecule LL37 itself, and who knows, one day we might be able to deliver a modified version of LL37 as a drug. That's the ultimate target. But before we do that, we have to understand what it does and how it does it.